Here's our last video, I mean it this time, gene and gene expression, the last section of chapter 3. This image really explains most of what you need to know. Let me talk you through it. When the gene is activated, enzymes come in and unzip the DNA and then copy the letters onto a new molecule called a messenger RNA strand. RNA is a close chemical cousin to DNA, and you might remember we get the materials for RNA out of the nucleolus. The key difference is that in DNA we use GATC, and in RNA we substitute a U for T. So GAUC, the law of complementary base pairing, or the ATCG rule, still stands. You just have to remember to swap out the T. There's no thymine in DNA. There is uracil in its place. That process of converting the DNA code into a short messenger RNA strand is called transcription. It takes place inside of the nucleus, and it is the first important step of gene expression. The messenger RNA, that blueprint for the protein, comes out of the nucleus and into the cytoplasm, where our protein assembly machines go to work, the ribosome. Now the interesting thing is the ribosome is also made of G's, A's, U's, and C's, and so the ribosome is also RNA. It's rRNA, ribosomal RNA. So we have messenger RNA, ribosomal RNA, and transfer RNA, not surprisingly made of the same G's, A's, U's, and C's. The transfer RNA has a unique code on the bottom that is complementary paired with the unique code on the mRNA strand. In this way, the code on the messenger RNA tells the transfer RNA which amino acids to bring at which point. And we're going to learn a lot about this in the coming week. But the order of the amino acids is critical to its shape and its function. There are 20 amino acids. We'll talk about them in a little bit. But for now, we'll just use colors. So this one is red, yellow, purple, green, red, blue. That's the order of the amino acids, and that will mean the difference between it being a waterproof protein or maybe a soft, smushy protein, or the difference between it being a tube or a keyhole, the difference between a receptor and an enzyme. So in this way, your genetic code tells your body how to fold your proteins to make you into the person that you are. The transfer RNA have 3 letters on the bottom called an anti-codon, G, C, U. And on the other end, they will bind to only one of the 20 amino acids. So this tRNA always has the same code on the bottom, and it always carries arginine. This different transfer RNA has a different code on the bottom, and it always carries aspartic acid. Each form of tRNA can carry only one of the 20 amino acids. It just so happens that there are sometimes more forms of tRNA that can carry each amino acid. Sometimes as many as 4, maybe 3 or 4 that carry aspartic acid. The point here is that the 3-letter code on the messenger RNA is the codon, and the 3-letter code on the tRNA is the anti-codon. Notice that anti has a T in it, which can help to remind you which molecule it's found on. The ribosome then takes the amino acids and hooks them together. Okay, so let's go through this a bit more logically. Here's transcription. The enzyme, not shown here, unzips the DNA and then brings in those complementary nucleotides, A's with U's on the messenger RNA, and C's with G's. This transcription starts at the promoter region, and the enzyme that makes the messenger RNA molecule is called RNA polymerase. ACE means it's an enzyme. A polymer is a long chain of little pieces, and it makes this molecule, RNA. Transcription stops when the polymerase encounters the terminator sequence. This is the stop sign that says, hey, stop here, that you're done with your recipe. If you go beyond, you're going to be into the next recipe. Watch this video, it's a really good one. Once the messenger RNA is released, it flows out of the nucleus, where the ribosome uses it as a template to build the little origami folded protein out of these amino acids. And so it's ultimately the order of the amino acids which will determine how the protein folds. And this whole process is called translation. Here we can see again the three forms of RNA. The messenger RNA, this one here, the green strand. The transfer RNA, these little orange trees. And the ribosomal RNA, don't forget the ribosome is also RNA. Through codon, anti-codon pairing, the ribosome receives the first of the amino acids, this green one called methionine. Then, reading the next three-letter pair, the next codon, C-U-G, it matches it up with the next anticodon, G with C, A with U, C with U. So this one is GAC, and this one brings in a different amino acid. Let's call it phenylalanine. Now here's the beauty of the ribosome, people. It works a bit like an enzyme to lower the activation energy, allowing for a covalent bond to form between the two amino acids. This is a super strong covalent bond called a peptide bond. It then boots out the empty transfer RNA and moves down the line. And in this way you can see the growing chain of amino acids that will fold into a three-dimensional protein. Great video, walks you through all of the steps that you need to know and all of the players. That's about it, ladies and gentlemen. Let me go back really quick to this one. If I were you, I would try to take this picture and remove all of the labels. Make sure that I could label them. Then I would get a bunch of Play-Doh and maybe some pipe cleaner or maybe Post-it notes. And I would actually make this machine run. I would put G's, A's, T's, and C's on my DNA, and I would transcribe them into G's, A's, U's, and C's. I would have a big old Play-Doh ribosome, and I would work my way through it methodically. So that if I give you this code, G, A, U, C, G, A, you should be able to tell me the two transfer RNAs that come in. Here's what I won't do. I won't expect you to know that these unique three-letter combinations correlate with this specific amino acid. You can look that up online. There are actually 64 codons, and that would be just mean. I'm not going to make you do that. What I will ask you to do is to know that there are 20 amino acids and to know that the order of the amino acids determines the location of the hydrogen bonds and thereby folding the protein into a three-dimensional shape, which has everything to do with its function. And so it is not a coincidence that my genetic code tells my ribosomes to make a special protein that looks blue in my eyes, and so my genes determine my eye color. Great work, everybody. I hope you have enjoyed today's video, and I'll see you in class.